1 1 1 1 1 1. Okay, we are back. Day one. And what we learned so far? So far, we learned that we were doing it wrong. Where's my notes? Oh, maybe I lost my notes. No. So yesterday we went on a tangent of chasing stuff and trying to go deep and learn random vocabulary without really agenda, and that doesn't work. It's just like jumping from one island to another one. And my OCD brain obviously excited about everything, so we'll try to bring a little bit more structure today, and we'll do in iteration. In the software development, it would call kind of agile. So we're not gonna, we are still against the traditional one, traditional education style. Me personally, when you actually go chapter by chapter, week 1, week 2, week 3, and basics and advanced, or maybe like chemistry, molecular biology, and so on. I would like bring try agile software development practice methodology, right? So we have a waterfall, agile, and then we have trying to find the image with the I didn't spell agile correctly. So yeah, and by the way, like this is great. That's what I want to bring. So this is traditional study, waterfall, right? So you learn what is wheel, you learn how they spin, you learn a frame, and then you get a car. This is agile. You learn how to ride the skate, then was that scooter, or I guess bicycle, bike, and then a car. Maybe we will experiment with this one. This is so cool, actually. Oh my god, the bottom one is great. So this is your prototype with like all the functions, and then you stream down functionality more, and you get the working project. So I think we'll go with this. We will go from kind of top perspective of big picture, like where biology, cellular biology stands right now, where's the gaps, what's the questions not unanswered, and where the research is, and also the science itself, like what does it mean to be scientists, like what they actually do. And is this a Lambo? what is what credentials do we need, right, to be counted as a scientist? Like, do you need a certain amount of white papers or peer reviews and so on? But this is cool, whiteboarding. Like, we might do whiteboarding today as well, because we need to run some ideas. I'm sorry. This is great. A great, great. What's the name? Meme. I'm gonna post it on LinkedIn. Anyway, so that's been said. We actually stopped on the question number 2 out of 50. We calculated that I need 46 years to learn biology. With that approach, I think I will go really quickly just to see my gaps, just to finish this one, and then we will go into research and see how it's like, and then we'll go into research with Agile. We basically try to learn a border between technology, software in particular, and biology, and trying to interline the what similarities and where the software can help modern biology. And we actually gonna start new chat with exactly this. I want you to do a deep research on the current state of cellular biology and where it intertwines with the software. What are the gaps? What are the hardest problem or most common problems that startups already, or companies, commercial companies, or maybe government already doing R&D, research and development? What are the areas that's still in the dark? What are the gaps? What's already useful? And how the AI applies to the modern cellular biology? Give me the list of areas, ideas, maybe startups, who is working on what. And most importantly, give me the list of latest and greatest studies or publications, so I can also learn, read it by myself, and get familiar with the current state, where we are as a humans being standing right now in the intersection of two spheres or industries of biology, particular cellular biology, and software or technology. Maybe robotic, but to be honest, I'm not a fan of robotics, so mostly software. Hold on. Deep research, okay. Okay, and now while it's gonna do deep research, we're gonna finish this quick. Oh, I see what's happened. I need this science. I think this one, right? Yeah. So question number 3, we're gonna answer those questions. What's the function of mitochondria? Bonus, if you remember the famous phrase about them. Oh, mitochondria, it's a energy power for the cell. So it's basically the vital, forget the name of it, vital part of the cell that produce energy, maybe ATP or something like this. How it's doing it, I don't remember. I remember it looked like zigzags or like a lot of membrane inside of the mitochondria. Mitochondria is one of the vital organelle of the cells. I think it can be many mitochondria inside of the cells, not just one, but many. And the more of them, the better. It produces all the energy for the cell and for the living organisms. The health of mitochondria really determines the whole state of the system of organism, how healthy you are, how well you feel, how much your energy, specifically in the human. It really improves recovery and basically everything. I listened to that podcast, and please find the reference for that podcast from Dr. Huberman about mitochondria, and they give amazing talk. Or they talk about it as a powerhouse for the power unit for your house. And basically, if 70% of your power suddenly dropped, like what can you power in your house? Pretty much nothing. Your quality of life will drop dramatically. So the health of mitochondria in your cells will increase the quality of your life. Bonus, if I remember the famous phrase about them, I don't remember. I don't even know that phrase. And I also don't remember what it's made energy from. I would guess from sugar or from a fat. Ketones make it from a fat. Mitochondria make it from the sugar. That's my knowledge on day one. Let's see how wrong I am, and let's see how much better that my understanding will be by day 100. I think last part I can drop, right? Yeah, that was for the stream. Mitochondria. How do I pronounce it correctly?
Mitochondrial.
Oh no, really?
Mitochondrial. Митохондриальный. Mitochondria. Mitochondria. Mitochondria.
I was pronouncing it so off. Okay. This is a strong answer. You're underrating yourself. Let me mark it up. Damn. Energy source for cells. Correct. Produce ATP. Correct. That's exactly the energy currency. Yes, that's the money, basically. The exact membrane inside. Correct. Those folds are called crystate. Okay, good. We have another vocabulary. Crystate. Okay, it's time to actually add those vocabulary into some kind of quiz, so it can quiz me. So since we in a browser a lot, widescreen, let's I term new CD code. Dear, what's that? School biology. Am I spelling it right? Biology. Oh no. Let's make it I'm going sidetrack again so horribly. New, but I like to work with AI, but I would like to work it consistently. So I need to set up these tools for myself. Otherwise, I'm going to be frustrated. So what I'm going to do, I'm going to create a repository visibly. I don't think anyone needs this visibly, but why not? Choose visibility public. Describe with you create repo. So now we're gonna clone it. Biology. Okay, git clone, perfect, cd biology, and now we're gonna start the Claude CLI. But I think I'm gonna start it in a open. In ideas that's not maybe ready made for it, but I like it. VS Code probably, whatever. Code, open, new window. Here we go. Let's start. Move to tab right. Right tab. Yes, I trans this folder. Close it. Close it. Okay. Flash init. So it will create a Claude folder where I'm gonna store commands and everything. I really need this just so I'm tired. Day number one, I'm already tired of this. Like, come on. Not analyze, repo clone option, at minimum clone, what is right? Yeah, I create a Claude.md, also create some skills. Skill number one, sorry, not skill, command. I need you to create a local command that I will trigger through the slash. And that command, I will put the keyword or vocabulary that I want to learn. You will save it into MD file. You will also organize this directory for learning purposes. I'm going to use it as a library for myself to learn biology. And we're going to use couple commands. One of them will be research, another one will be quiz. So I would say slash vocabulary, right? Or slash remember, and will give you the word. You will put it into the list, and then you're going to quiz me when I'm ready. When I type command slash quiz, we're going to do a quick quiz, like 10 questions. And you will keep track how well I know those questions. Let's start from that, as simple. I'm sorry. Okay. We have a repo. All good. So those faults are called crustate. Yeah. I do need a tool to know how to pronounce it.
Christe.
Crustate. What is the best tool? Let's use Perplexity. I don't have a lot of credits, but I'm learning biology and a lot of vocabulary I don't know how to pronounce correctly. What is the best tool for macOS or maybe online where I can put a word and it will give me clear pronunciation, maybe slow, fast, so I can learn how to pronounce it. If the tool can listen to me and give me feedback, that's even better. Okay. What we have here? Yes. Yes, let's go. We're gonna wipe code it. We're gonna wipe code this study. Do you want to make this edit to vocabulary MD into which command? Yes. Yes, I do. Yes, I would like that. Research vocabulary, Claude, quiz. Love it. Okay, it's even gonna track my statistic. I decided just to finish 50 questions to see my gap, just to finish something from day zero. And like with 30 minutes in, I already do 3 different things. Setting up repository, learning for the tools. Good luck. And here's the things, like with this small attention, like today, everyone on a scroll, right? And like those shorts video, TikTok, YouTube, Instagram, that small attention disorder that everyone have now. Maybe not disorder, that's the way heavy. Not like the way heavy tune up our brain with this motion and quick dopamine signals. It's so hard to stay in one topic. Like it's really hard. It just, for me, it's super hard to read the article. Like I'm reading first paragraph, I have three different ideas. I'm open up, like I have 3 monitors. It's insane. And I have no idea how the kids at school even handle it. Like if I have this problem, the kids has that problem multiplied by 10. And for them to study, to sit like 20 minutes, 30 minutes in one subject and listen to a professor saying something, I don't know, that's probably so hard. Respect to kids. What is this? I'm learning my biology a lot. Oh, that's a perplexity, right? So we closing this outage. I remember this phosphate, yes. The phosphor and O4, PO4, PO4, PO4. Mm-hmm. State. Okay, we got our tool done, file created, blah, 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 vocabulary. Oh, here we go. Voice. Toggle voice mode. Oh, I can do voice? I need to restart this session, I think. Terminate. Yes. Okay, I add this to my vocabulary. Probably spent like 1 million tokens for this. I don't know why. Just add it. Like, why you do that? Let's see if it added. Yes, don't ask me, like this is so vocabulary. Damn, I like this. So it will create a file for each of them. Etymology, Latin. I love this. Oh, then we're gonna write a script that will pull images to each vocabulary, so it will show me images sometimes and sometimes word, and I have to explain what it is. Yeah. See, I switch again. Those faults are called cristae. Cristae. The folding increases surface area. Yes. More membrane, more energy production capacity. So basically, it's like in your brain, right? Like the more folds the brain has, the smarter you are, I guess. I don't know. I have each to ask that question now, but I'm not gonna do it. We need to finish this. Many per cell, not just one, correct. Cells with high energy demand, muscle, heart, neurons have 1,000. Oh, what that's I didn't know. I had no idea that. Red blood cells have 0. Interesting. Why? I have so many questions. Why red blood cells have 0 mitochondria? And what was another question? Many cells, not just one, correct. Production. I also had a question. Surface area. I don't know how to spell it. Okay, disregard the question. Mitochondria. I had no idea red blood cells have 0. More mitochondria, more energy capacity, correct. And this is exactly why endurance training works at the cellular level. Oh, interesting. It's trigger your cells to make more mitochondria. Biogenesis. I like this. Biogenesis. Vocabulary. Biogenesis. See, it's faster now. It's actually create a library of those vocabulary and it will quiz me. Health implications, correct. And this is generally frontier research right now. Mitochondria dysfunction is implicated in aging. Yes, we're gonna be young if we do it right. Neurodegeneration, metabolic diseases. So that could be a goal, like how can we increase mitochondria, like simulate mitochondria growth, right? Imagine if we create a AI model that will simulate how mitochondria will, the growth of mitochondria will react in a certain environments, organism or environments, and maybe even pills, right? Supplements. And is it only chemical, right? Maybe not only chemistry stimulates the growth of mitochondria, maybe something else. And that's what we're gonna discover. What? That's a good paper to read, by the way. we do have research, right? Give me the list. Okay, yeah, I do deep research. Still, I have to like write a software right now that will track how much I switch the topic or subject. Like I can imagine maybe every 5 minutes, maybe even shorter. My attention span maybe even shorter. Why did I switch to the research now? I didn't even finish answering, like reading the answer of this question. Correct. This generally frontier research right now may implicate aging neurodegeneration. I like this metabolic disease. Metabolic diseases. I'm sorry, something off with my voice. I guess my mitochondria need to take care of my cough. Energy from sugar. Correct. Primary glucose. Interesting. So in a ketosis, mitochondria is struggling. I really want to know now how the ketosis and ketogenic diet will affect mitochondria health and amount. Can we even measure how many mitochondria? Is there a test? Is there a test? Startup idea number one. 30-second test of amount of mitochondria you have. But we cannot do it from the blood, right? Because we just learned that the red blood cells doesn't have mitochondria. So you have to take a tissue, like a skin. That would be insane. Is there a test? Is there a easy test to measure mitochondria count? By the way, LLM measure. Is there what? Measure? LLMs make us dumber every single time. Bonus also from fats. Oh, correct. Primarily glucose. Bonus also from fats. Fats acids and even amino acids when needed. Your ketones. Oh my God. I couldn't even finish a paragraph and I already jumping into different researches. That's exactly what I was trying to research, but it's already answering. Your ketones not is the right to. Ketones are an alternative fuel made from fat that mitochondria can also burn. Okay, now I have to clarify this. But I'm not going to do it right now. I have to put it here. The famous phrase, mitochondria are the powerhouse of the cell. That's what I said. You basically said that, yeah. Powerhouse, like power unit for your house. That's what I said. Power unit for your house. That's a powerhouse. I see. So I'm giving you full credit. My first credit, yeah. Day one, we got one credit. How many credits do you need to graduate? Tweaked. How they make ATP? Since you said you didn't remember, sugar gets broken down in steps. The final step happens in the mitochondria via the electron transport chain. Love this. I don't know what it is, but I want to see it here. Oh my God. How can you study linear if it's so cool? I have to print this.
Welcome to the next lesson in the Dirty Medicine Bio.
Dirty Medicine. Thank you, Dirty Medicine, for this. Can you give me this screenshot? I really like that. Copy image address. Oh yeah. Electron transport chain. This is how sugar converted to ATP. Let's see how much I understand this. I understand this electron. With K, that's almost like my initial. Q, O2, O2 minus, no idea. Some kind of photons going out or electrons going in. No idea. State C, stage 1, stage 2, 3, 4. And here's our ATP going out byproduct. ATP synthesis, ADP plus PI, hydrogen, hydrogen, NAD, and yes, this stuff you can buy on Amazon. That's what I know. NADH. Okay, I'm printing this. B. Yes. Like when you study, something have to keep your excitement up. Sitting on those folded crystate membranes, it uses oxygen as the final electron acceptor, which is why you breathe. Every breath of O2 you take is ultimately for your mitochondria. Oh my God, I had no idea. I thought that's for other oxidation processes, but I had no idea this is for mitochondria. I thought like DNA replication uses, or I literally didn't know that it's part of the fuel for mitochondria. CO2 you exhale is a waste. I won't search for that specific Huberman episode unless you want me to. Let me know. A score 1 out of first full point. Yeah, strong domain. Running tally. What is tally? What is tally? Tally. But wow. Yeah, English is not my first language, so sometimes I need to translate it into, like, I'm learning English as well. Pattern immersion, you have pop, science, podcast level, conceptual knowledge. Oh, they already have immersion patterns. That's I have pop science and podcast level, conceptual level. You understand what things do and why they matter, but gaps in vocabulary, yes, and molecular mechanics, yes. That's exactly right. Like, it nailed it. Specific names, the specific chemistry. That's actually a great starting profile. Concepts are harder to build than vocabulary. Oh, thank you. A little of motivation. What is a gene? Gene, you can answer it at any level, wagging to your shoulder. Okay, gene, it's a representation of the whole genome, and it's a small chunk of DNA amino acid chain. I remember it's not protein. And each gene responsible for, the scientists used to believe that each gene responsible for a certain function in your organism. So it's basically the color of your eye, turn on or turn off, certain behavior of your system. And I don't remember the size of the gene. I would say it's a 3 base molecular, so it's a 3 pair of combination. ATGC. So any pair, like any three pair, it's a gene. That's what I would guess. I also don't remember how many genes we have in the humans. I know the simple organism like worms and flies has less gene. And I know that human genome was sequenced a few years ago fully for the first time. But I don't know what that means. I don't know what sequence mean in that case. Recently, with all that AI revolution, I feel like genes, it's actually weights of the model. I don't know why I'm making this comparison. Maybe it's enforced by social and public opinion. But feels like, because when you look in the model and its weight, it's kind of useless, right? Unless you pass a lot of information through those weights. Otherwise, it's just CSV file or just like literal weights for every neuron. So feels like genes and genome and DNA works very similar. Basically, it do nothing until replication happening. And I know that we used to think, when I say scientist, or maybe me personally, or humans, that genes not changing only when sexually reproduced. But right now, I think the genes can change, and this is based on there is DNA and there is I forget the word. Maybe it will come to me. Apogeny. No. Yeah, I don't remember that word. So during the life of the system organism, the genes can change, not just by mutation, but epigenetics. Yeah, like I think epigenetics, it's ability of organism to turn off and on certain genes during its life, not only during the reproduction. While it's translating it, I'm gonna get my picture. I want this t-shirt. Whoever made this image, full credit to you. I think it was a YouTube channel or something, right? Dirty Medicine. Good job. Maybe it's not them, but I think that's what I clicked. And I definitely need a t-shirt like this. Let's see what my answer is. Oh, I didn't even click it. This is really rich answer. You are doing the things good learners do. Connecting ideas across domain. Yeah. Okay. Learners, remember, connect ideas across domains. The problem when you're young, you don't have any domain. That's why to connect them, it's hard. But every single year, every single day, when you learn new, that's what expands, and you have more stuff to connect to. Across the map. By the way, maybe this is all fluff and just giving me props for no reason. The AI weights analogy, surfacing forgotten vocabulary, myth, thought, epigenetics. Yeah, I did that. Yeah. And being explicit about confidence, let me mark it up carefully because there is a lot here. Small chunk of DNA. Yes, exactly. A gene is segment of DNA, not a protein. Yes, correct. Genes are made of DNA. Huh? Oh. So it's not DNA made of genes. It's a gene made of DNA. They often code for proteins, but they aren't proteins themselves. That's true. Responsible for a function, eye color, turning behaviors on off, correct in spirit. One gene, typically one protein. That protein does a job. Eye color enzyme, and I don't know how to pronounce this. Ferment.
Enzyme.
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 Enzyme. Eye color, enzyme production, hair texture, etc. Simple organisms have fewer genes, generally true with weird exception. Some plants and amphibians have way more genes than humans. Oh, that's a vibe coding. That's vibe coding. That's what it is. That's biology vibe code. Like, it creates way more gene than needed for this car. Yeah. Okay, biology explain. We finally know why amphibians and why some plants and amphibians have way more genes than humans, because they've been vibe coded. Okay, I need to do a post about this. Generate a simple LinkedIn post. It's a meme and fun. So it's basically humorous post. The hook would be, we finally know why some plants and amphibians have way more genes than human. And the answer is because evolution was playing with evolution was vibe coding. Vibe coding? No, vibe. Vibe coding. Okay, we 1 hour in. I'm on the second question. I don't know what to do with me. Oh, I can't study. I just cannot focus. Like, I need to study where technology, software, and biology intercept. And I'm in the question number 4, answering what the gene is. But what I learned today is that red blood cells doesn't have, oh yes, that's what I need to do. Another to do. Twitter and what I learned. Or X, right? Human genome was sequenced, correct. First full draft 2003 truly completed telomere to telomere. Oh, I know what telomere is. I think it's the ending of DNA. And with every single division, one telomere molecular, I guess, becomes smaller to the end that's at certain point it exhausts itself and DNA couldn't divide anymore. And the visual that I have representation in my brain is like shoelaces, and at the end of shoelaces we have those I don't know the name of that stuff that protect of material to go haywire. But I would like to add it to the vocabulary. I need to change that command to vocab. Epigenetics, correct. Genes can be turned on and off during life without changing the underlying DNA, often via chemical tags on the DNA. Oh, so basically, some chemistry working on a DNA, I mean a chain. I hope I'm saying it right now. And it's turned on and off DNA gene, and when protein created from that gene, it's basically looking if it's on or off. That's what's up. Often we are on DNA or on the proteins DNA wraps around it. I'm lost, but I'm not gonna dive into it. Diet, stress, exercise can influence this. Okay, kids. Physical activity, it's important. Some epigenetic marks can even be inherited, which broke another scientific dogma. Oh, so epigenetics mark can be inherited. I think that what, okay, I think I know. I think that's what's happened when they got mice, and every time when they give them a smell of orange, they shock them with electricity. Classic science. I was asking earlier what scientist does. The scientist shock mice with electricity. That's what's up. So smell of orange, shock. Smell of orange, shock. It's basically created a reaction on the epigenetic that they remember that, and then they remember it so hard that it's even affected their epigenetics and turn on or off certain genes. And when they reproduce sexually, the sibling, not a sibling, the new generation, when they smell orange, they were freaking out. They were freezing. They didn't know what it is because they never experienced shock of electricity. But the epigenetic was telling them like, shit, that thing's gonna go to shit. So they were freezing. They experienced some feelings that didn't know what it is because it was from the previous life, from the lives of their parents. I think I paraphrasing that experiment right. If you know what it is, mention it in the comments. I will read the original version. Representation of whole genome. Ellipsis. The genome is a whole set of all your DNA, all your genes. A gene is one small piece of the genome. Genome is the whole library. Gene, one book. Okay. Representation of the whole genome. Genome is the whole set of all your DNA. I'm confused. So how many? Amino acid chain. This is the protein versus DNA mix up again. Same on question 2, words wording it. DNA / genes, chain of nucleotides, not amino acids. Nucleotides. Okay, I'm gonna read, yeah. Proteins, chain of amino acids. Oh, yeah. 20 different ones. There is only 20 amino acids? There only 20 amino acids? Acids? Interesting. Genes are instruction for building proteins, that's I know, but they are written in nucleotides language, not amino acids language. Yes, got it. So proteins is written in amino acids, and DNA or genes read in nucleotide. The three pairs is a gene. Close to real concept, but misapplied. Three nucleotides in a row, a codon. Oh, codon. Vocabulary. See, it's easy, like with this. And now we have like all this library, and then we're gonna throw Karpagi Obsidian knowledge graph of this. It's gonna be cool. Codon.
Codon.
I mean, yeah. Which codes for one amino acid? Okay, now I'm confused. So three nucleotides in a row, a codon, which codes for one amino acid. So three nucleotides is one amino acid. A gene is typically hundreds to thousands of nucleotides long. Enough codons to spell out entire protein. So three nucleotides, one codon, one amino acids. A gene, many codons, a whole protein recipe. I need to get this. I need to dive into this, maybe offline. I have no, like, I don't see a clear vision of this. Maybe next time I will try to draw it. Don't know how many genes humans have, roughly 20,000. Protein coding genes. The shaker. That's about the same as tiny worm. C. elegans has 20,000 too. Complexity isn't really about gene count. It's about how genes are regulated and combined. Wow. The AI weights analogy. Oh, little fire, yeah. This is actually a legitimate good analogy, and biologists / ML people use it. Genome equal it trained weights. The static instructions, the cell machinery reading and expressing those genes equal inference. I should know how to pronounce this word.
Inference.
Inference. Equal inference, yes. That's exactly what I mean. Pronunciation is lacking. I mean, I know. The dynamic process that's produced behavior. Basically, what we're doing here, the model goes through the inference, taking my input tokens, going through the weights, and outputting next and next token. So producing I or IN, F, maybe there, it's one token, right? that they've been produced through the inference of the model of Opus 4.6 extended printing. Dynamic process that produce behavior. Your instinct that it does nothing until replication happens is half right. Actually, it does nothing until expression happens. Transcription plus translation, which we will get to. Okay, replication is just copying the weights. Expression is running the model. Okay. Oh, okay. Replication is copying model to another VM, let's say, right? And expression is running the model. And expression, this mean it will create proteins. Maybe. It will inference, read it, and create protein based on it. Hold on this analogy, I'll keep paying it off. It will keep paying it off. Yes. Sequencing. Since you flag not known, it just means reading the order of the ATGC letters along the DNA, like reading a book, letter by letter. Sequencing the genome, reading all 3 billion letters in order. That's I'm not sure I understand. I mean, the order is different in every human. Is it right or not? That's my biggest question for today. The sequencing of human DNA. They're sequencing the genome, reading all 3 billion letters in order. My question is that order same for every human being? Or it's different? Since everyone have different DNA, or we have different gene expression, I guess, right? But the order is the same. So what's changing? Like on and off switches changing, but the order stay across human being as a species consistently, or the order is changing? And the expression of turn on and off of some genes, or maybe even codons. I have to ask this question. New chat. Project. I need to clarify that right now. Oh, great question. You are circling something real. Let me untangle it. The order is almost entirely the same, almost entirely, across all humans. That's actually what makes it possible to talk about the human genome as a single thing. Okay, so the order is the same. Any two unrelated people are about 99.9 identical. Oh, so it's almost mean it's never absolutely the same unless it's a clone, but it's 99.9. Okay, now it makes sense. So any small changes in the order makes us different. Is that what's identical at the DNA letter level? The 3 billion letters comes in the same sequence, in the same order, on the same chromosomes, with the same gene, in the same. Okay. What? I need images. I need images for this. Like I'm losing it. What difference between people is that 0.01%? Small variation, sprinkled throughout. Mostly these are single letter swaps called SNPs, where you have an A and I might have the G as the exact position. These are also small insertions, deletion, and some large structural variations. These differences are what make you genetically distinct from me. Eye color, diseases risk. Okay, so they basically, in those 3 billions, identify which gene represents the eye color, and they basically flipped enough SNPs to basically get a brown, black, blue, green, yellow, right? Am I right? This is a small insertion deletion, the different makes you caffeine. I need some caffeine. This difference of what make you genetically distinct from me. Eye color, disease risk, how you metabolize caffeine, etc. But the overall sequence and structure is shared. Now, to your gene expression intuition, yes, that's a separate layer, and you're right to suspect it. Every cell in your body has the same DNA. A neuron and a liver cell are identical at the sequence level, but they look and behave completely differently because different genes are turned on and off. That regulation happens through. Oh, yeah, what? Back to English lesson? English classes 101.
Through.
Through. Okay. Through. That regulation happens through transcription factors, proteins that bind NDA and switch genes on and off, epigenetics, chemical tags like methylation, added on top of DNA that don't change the letters but affect whether they get rid. That's it. There is 4 dimension. 4. What is it? 4 dimension. Number 1, order. That's what makes us a little bit different. And if order change significantly, then we become a different species. I'm assuming. Maybe there is 5 dimension, like amount of genes, then order of genes, then I think I lost 1 dimension. The order. I'm trying to introduce dimension as a variables, what will affect the expression of cell. Amount of, I'm not sure about amount of genes, right? So let's keep it what I already comprehend. Order, and then each genes also going on and off. When it's reading it. And third dimension, it's a chemical tag. So alternation of weights at real time. Let's take it with this 3. And if we keep the analogy of the weights in a LLM model, then the order of the genes would be, or amount of the genes, right? It would be how big is the model, right? How many parameters? 3 billion, 1 billion, 1 million, and so on. And then the order is that like each company probably defines the LLM model with different layers, so we can consider that as the order. That's why we have humans and dogs, and that's why we have Claude and GPT. Yes, I think the order would represent LLM architecture, not architecture, the actually neuron architecture in that neural. That's number one. Number two, what's getting read and what's not getting read, that's epigenetics. And epigenetics, that's probably post-training. It's basically force them to turn on and off sometimes. And sometimes feedback propagation can be considered as the epigenetic, maybe. And the last one, the chemical one, chemical tags, that's actually fine-tuning. Epigenetic may be fine-tuning. Fine-tuning is the epigenetic, let's put it this way. Post-training / fine-tuning is epigenetics, and the chemical tags, that's our prompts, maybe. Maybe it's actually fine-tuning. So it's basically altering weights at real time, but not altering physically weights, but it's basically saying like, don't even go to this directory. I want you on purpose to turn it off. Interesting. I want to keep this. I want to keep that idea, and I know it's out of blue, or maybe it's not. Maybe a lot of people are thinking exactly the same way. But I want to connect this as much as possible until it's breaks. In terms of analogy, non-coding regulatory regions, stretches of DNA that act like dimmer switches for nearby genes. Wow. I have huge respect for all the scientists who got us to this point. Like, I have no idea how much hours, lives, dedication, madness has been put into this in order for me right now to get that knowledge by just typing 3 letters. Like, thank you. This is amazing. Like, this is years of work. We standing on the shoulders of giants. 100%. Moving on. So oh, how deep I am. I'm still answering the second question. So expression vary between your cell types, between you and me, and even with you over time. That's a new. I knew that expression varies because expression, it's an accumulation of all these 3 variables or dimensions. I didn't know if we already talked about. Codons are different things again. A codon is just a 3-letter chunk of DNA RNA. Okay, what is RNA? Where is my vocabulary, right? RNA. That code for amino acids, the codon code itself, which triples means which amino acids, is universal across near all life, not just humans. Okay, interesting. What can vary is codon usage. Sometimes multiple codons code for the same amino acids, and different organisms or even different genes prefer different ones, which can subtly Back to the English lesson.
Subtly.
What?
Subtly.
There is no D-E-N-A here. Subtly spelled like this. Subtly.
Suitily.
Subtly. Okay. I'm doing spelling bee now.
Subtly.
Subtly, exactly. So,
Subtly. Subtly.
subtly.
Subtly.
And this is subtly. It sounds exactly the same. Wow. English is strange. Which can subtly affect how efficiently a protein gets made. That's very interesting. So, what is protein then in the models, right? Or even in the coding and software? What is protein in the software? That's I need that. I need that answer. What is protein in so? So to diversity answer. Yeah, I got it. So thank you, Ross. Score 0.6 sequence expression happens on score since you flag that's real order of the ETG on DNA update podcast. Hold on. Where did I stop? Using that doesn't nothing until replication. Yeah, expression, inference, replication, just copy and paste, keep sequencing, like reading a book. I still, yes, I know what sequencing, but I need to know which human actually been sequenced. Yep, I do need to know now. Score the strong conceptual gaps, vocabulary slips, DNA as amino acids, chain mix-up happens twice, so I'm flagging it as your number one gap of fix early. Thank you for flagging it. I have a mic here, just in case. And I don't think it's used, but thank you for fixing it, for flagging it. Running tally. I learned what tally is. Update profile, podcast level content fluency, beacons, the chambers vocabulary layer. Same pattern, good news. The conceptual layer is the harder one to build, and you have. Okay, now we need to create a profile for all students who are studying right now. What are their profiles? Like, personality types. That like Myers-Briggs personality types. Like, where's my image? Give me image. Copy image address. We have 16 of them, right? Yes. And now we have a study or learners types. And no, that's not like visual or audio and so on. Like, I think Veritasium proves that we all think that we are visual, but no. And for learners type, that's what we need to create. I think a lot of people right now learn and they have knowledge of podcast level conceptual fluency. Like, this is very interesting because majority of my study was from a podcast. That's so true. And like in the models, like the shitty data, shitty models, right? It's very important that podcast that we listen, it's actually truthful. profile, podcast level update, something here. I like that. Like something is here. I feel like we're gonna create a chain. We're gonna create a workflow of everything what's resonate with me should go into some kind of pipeline and process a little bit further. I don't know why, but maybe. Same question about, wow. I don't know, guys, I'm broken. Second day, and in the beginning of day number 1, I noted that the way what we done in a day 0 was wrong, and we should not repeat it, because for 2 hours we answered 2 questions, and now 90 minutes pass, and we answer 2 questions. Loosely related, question number 1, loosely related to where you just went. What is the central dogma of molecular biology? If the phrase is unfamiliar, guess from the world, certain dogma tells you it's the main core idea of the field. I don't know the answer for this. I'm gonna fully guess. So the dogma is, I don't know what's the dogma, but molecular biology, it's you can achieve same proteins or same results from different part of codons, maybe. Also, the way how your cells build the proteins heavily varies based on multiple parameters or variations on a different dimensions. Number 1, chemical on-off switches, then epigenetics, order of the genes or sequence, and maybe something else. So to basically study molecular biology, it's so hard because it's just crazy amount of variation, and we need huge amount of compute to actually run through all single variation and find local minimals or results that we did not see before. Just basically to brute force, it's crazy amount of compute. And before we were just guessing it or poking around to see what comes out. And that's not gonna take us far. We either need to build a pattern or to brute force it, but to brute force it, it's a lot of resources and compute. I don't think we have enough for now. Or maybe we do now with all these AI data centers or compute centers. But we definitely need new chips. Or maybe we will use a software how we can use the molecular biology to actually run some GPUs, because GPUs already exist, and now we're gonna do inference of cell expressions or protein expressions, DNA expressions. I don't know what other words I'm gonna throw there, but we're gonna do inference or expressions of DNA genes on GPU level. Therefore, we can finally simulate how the cell behave under different medicine, different organisms, different environments, and so on. And that's what we're doing. I mean, thank you for compliments, AI. I love you. This answer is fascinating because you don't know the content of the central dogma, but you intuited the things that's model for. I have a hard, I think it's compliment, but I don't understand what that means. The information flow problem in cells. Let me give you the answer first, then mark up your guesses. The central dogma of molecular biology. It's the foundational model of how information flows in cells. Yes, the way how information flows in cells, that's what's up. But what's dogma mean? Dogma. Is a principal belief or set of doctrines laid down by authority, such as church governmental group that's come. Fundamental foundational beliefs or established tenets, I don't know what's tenets, accepted as uncontrovertibly true. So it's basically axioms, right? In geometry, maybe. Mhm. Where am I? Oh yeah, okay, got it. So the information flow problem. It's the central dogma of molecular biology. It's the foundational model of how information flows in cells, proposed by Francis Crick, one of the DNA double helix guys, in 1958, in one sentence. DNA, RNA, protein. That's it. That's the dogma. 3 steps. DNA, RNA, protein. I'm so excited. I don't know why. And remember this. The t-shirt have to be made. It's called DNA to RNA is called transcription. The cell makes a temporary portable copy of one gene by transcribing it from DNA into similar model called RNA. Specifically mRNA, messenger RNA. Think photocopying one page from a giant reference library you can't take out of the building. One page from a giant reference library you can't take out of the building. Okay, so basically if your agent use only a particular niche of the model, certain weights, instead of downloading 100 gigabytes of the model on your machine as a DNA, we can get a RNA, a small copy, just for your, not general knowledge, but for your niche knowledge will get RNA to local machine. Since it's smaller, it needs smaller memory, smaller compute, and we can run it against it, and our inference will be faster. That's next steps. That's next step in AI. We have to invent RNA for LLMs, the messenger of small chunk of your particular agent. So when you send the prompt, it needs to fine-tune which weight it will use. Am I right? We don't need all weights. That's all I'm saying. We need only chunk of it. I don't need the knowledge of Saturn if I'm writing a marketing email about garbage disposal to the Bob, right? So I just need the RNA. Give me that. I'll do inference locally. Your data centers are like, ease it up a little bit, and then just do it locally. Like it's like JavaScript executes in browser, right? It doesn't execute in server. It's like my CPU will do that. My GPU will do it. We need RNA. We need new protocol. RNA for LLMs. That's what we should do. Your AI weights analogy. Oh, I didn't even read this. Oh my God. I need to read before saying something. Think photocopy, yes. So RNA protein is called translation. The mRNA copy travels to ribosome. Do I have that in my vocabulary? Your AI weights analogy is the inference engine. The ribosome reads the mRNA in chunks of 3 nucleotides, codons, the things you almost named in Q4, and for each codon it adds the correct amino acid to a growing chain. When the chain is done, it folds up into protein. Easy. I'm so lost. So the full pipeline: DNA storage, transcription RNA messenger, translation protein, the worker that actually does stuff in the cell. Why dogma? Greeks use the word semi-jokingly. He means as a core principle, it's been revised since RNA can go back to DNA in retroviruses like HIV. Wow. There is so much to learn. Like what? They saying that messenger can go back to the storage. It's not one-way function. And that's what HIV is. It's broke one-way function. That's what you're telling me? Which is why dogma is now a bit misleading. Okay, thank you. But the basic flow is the foundation of literally everything in molecular biology. Yeah, I know that flow, but yeah, the dogma. This is the single most important concept in the field. If you only remember one thing from this whole quiz, make it DNA, RNA, protein. Okay. Your guess marked up. Same protein from different causes. Wildly, you stumble onto a real phenomenon called codon degeneracy. Redundancy. Redundancy is better. This word I need to know how to pronounce.
Degeneracy.
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 Is it me or she's saying sassy way?
Degeneracy.
Probably me. Redundancy. There are 64 possible condoms. Like, guys, we have 64 bits architecture, right? Or 86. 64. 32. 60. Yeah, 64. Intel. Oh my God, this is so similar. Like, what happening? There are 64 possible condoms. That's what we are running our compute for. I know all the comments. Stop connecting things that's not connected just because the numbers are the same. But that's what I'm doing here. I'm connected unconnectable numbers. Take it as a joke. There are 46 possible contents. 4 base 4 base 4. No, it's 2 base 2 base 2 base 2. But only 20 amino acids. Again, I see this again. 20. There's only 20 amino acids. Second time, I need to dive into this. So multiple codons code for the same amino acid. You weren't asking this, but you accidentally surfaced it. Worth a partial credit point. I got a credit point from building proteins varies based on chemicals, on-off switches, epigenetics, order/sequence. This is gene regulation, a real and huge field. You are describing why two cells with identical DNA, your liver cell and your neuron, can be totally different. They express different subsets of genes. Correct intuition, even if not the dogma itself. The compute, brute force, GPU tangent. This is interesting. You are not wrong that biology is computationally explosive. Combinatorial complexity is brutal, and there is a real and active fields doing this. AlphaFold, DeepMind. Yes! 2020 basically solved protein structure prediction. So the protein folding, I think what happening is it's not just depends on different proteins do different things, but also the way how they folds matter a lot. And compare that to the chemistry, we have different molecules and elements, but the way how they create a structure is different. So graphite and graphene, not a graphene. Anyway, sidetrack. You know what I mean. It predicts the 3D shape of a resultant protein. This was considered an unsolvable problem for 50 years, AlphaFold. EVO, ESM, RF diffusion. Newer models doing what you are describing, simulating biology on GPUs. Oh, interesting. Companies like Recursion and Citro isomorphic labs are doing exactly the simulate cells response to drug work you described. I do need to know this. I need a knowledge graph for this. So your instinct about where the field is heading is correct. You're close to current research direction than the central dogma, which is 1950 idea you weren't going to deliver from this first principle. But none of that is that central dogma. The central dogma score 2, credit, real adjacent concept, but you didn't have the action, 46%, but the numbers misleading. Let me update the profile. You strong intuition, mitochondria, genes regulation, AI, bioanalogies, where the field is going. Decent pop sci SCI exposure, Huberman genome sequence news. You like vocabulary where nucleotides versus amino acids, transcription, translation, codons, ribosome. The canonical foundational facts. I think I'm losing it. Central dogma, basic taxonomy, like prokaryotic, eukaryote, prokaryote. I know that now. Hey, I know that. I learned it yesterday. Implication for how to study. You probably don't need an intro to biology for kids. Thanks God. Approach. You need a vocabulary scaffolding path to attach the right words to concepts you already half understand. Then you can jump deeper fast, different from the linear textbooks. Oh, yes, plan I give you originally. Good question number. Remember, guys, there is 50 questions, 50, and we're gonna do them. Let's stay in the neighborhood since you are warm. What's the difference between DNA and RNA? Name as many differences as you can, even once is fine. I'm gonna answer that next day. I know the answer because RNA it's a small chunk of the DNA for not a transcription, transported. It's a DNA at mimic chunk of DNA, so it can be moved and then read. But I'm not going to answer it right now. Day 1 was successful. See you tomorrow.